Let's begin by looking at the process of fertilization as well as some of the mechanisms behind the specification of the different axes of the body and the process of cleavage. The majority of frogs have external fertilization in which the male fertilizes the eggs as the female lays them. It's worth noting that there are a few species of frog that use internal fertilization, and in that case the eggs are fertilized inside the female's body before they are released. Frog eggs have a distinct polarity before fertilization, with the vegetal pole having dense yolk and the animal pole having little yolk. In some species, the two poles also have a different pigmentation, so that the animal pole, as you can see here, appears pigmented. Another important difference between the animal and the vegetal poles is the uneven localization of proteins and mRNAs, as we'll see later. In amphibians, the sperm can enter the egg anywhere in the animal hemisphere, and the point of sperm entry is important because it determines the dorsal ventral axis of the body. The side where the sperm enters becomes the belly or the ventral side of the embryo, and the side opposite of sperm entry becomes the back or dorsal side of the embryo. The establishment of the dorsal ventral axis in the frog involves the rotation of the cortical cytoplasm. And recall that the cortical cytoplasm is the cytoplasm that's located right underneath the plasma membrane. That rotation of the cortical cytoplasm requires the microtubules of the oocyte to be reoriented, as you can see here, so that they are parallel to each other. And that rearrangement of the microtubules is achieved by the microtubule organizing center of the sperm. As a result of the rearrangement of the microtubules, the outer cortical cytoplasm is able to move independently of the inner yolky cytoplasm. In this way, the microtubules allow the cortical cytoplasm to rotate with respect to the inner cytoplasm. And the movement of the cortical cytoplasm sometimes exposes a region of the inner cytoplasm that appears gray. And that region of exposed gray cytoplasm is known as the gray crescent. That's located directly opposite to the site of sperm entry. The region of the gray crescent marks the site where gastrulation begins, and even in species in which a gray crescent is not exposed, the cortical cytoplasm still rotates relative to the inner cytoplasm. This means that the site where gastrulation starts is going to be at the side opposite of sperm entry, which becomes the dorsal side of the embryo. Recall from previous lectures that the pattern of cleavage in amphibian eggs is holoblastic. However, because amphibian eggs are mesolecithal, having more yolk in the vegetal pole compared to the animal pole, the cells at the animal pole finish dividing slightly faster than those at the vegetal pole. As you can see from this diagram, the first division begins at the animal pole and slowly extends into the vegetal pole. In species that expose a gray crescent, that first cleavage, which is meridional, passing through both the animal and the vegetal poles, divides the gray crescent equally between the two resulting blastomeres. Before the first cleavage has passed through the vegetal pole, a second cleavage that is at right angles with the first one and also meridional begins. Unlike the first two cleavages, the third cleavage is equatorial. However, because of the presence of more yolk in the vegetal pole compared to the animal pole, that third cleavage is not exactly at the equator, but is actually displaced toward the animal pole. As a result of that slight displacement in cleavage, the animal pole region becomes packed with numerous small cells that are known as micromeres, while the vegetal pole region contains a smaller number of large yolk-laden macromeres. The stage at which the embryo has 16 to 64 cells is known as the morula stage because the embryo looks like a mulberry, and the same is true of mammalian embryos. Once the blastocele forms, the embryo is considered a blastula. The blastocele is more than just a fluid-filled cavity. It actually plays important roles during amphibian development. One of those roles is to prevent the premature interaction between the animal and the vegetal pole cells. In amphibians, the cells of the animal pole will give rise to the ectodermal tissues, while the cells that are located under the blastocele will become the mesoderm, and those located at the vegetal most region will give rise to the endoderm. If the blastocele was not present and the animal pole cells were allowed to come into contact with the cells of the vegetal pole, the animal pole cells would actually become mesoderm instead of ectoderm. So the blastocele is important for keeping the ectoderm cells undifferentiated until during gastrulation the cell movements lead to rearrangements that place the cells in regions that will allow them to develop as ectoderm. The regions of the developing embryo that are located opposite to the site of sperm entry will give rise to the neural ectoderm, the notochord mesoderm, and the pharyngeal head endoderm, which is not labeled in this figure. As in other organisms, the mid-blastula transition in amphibians involves a decrease in the frequency of cell division, which results from the acquisition of gap phases in the cell cycle. Additionally, recall that at this stage, the genome of the zygote begins to be expressed, and that's achieved both via chromatin modifications and the demethylation of promoters, so the demethylation of DNA. This switch to zygotic gene expression involves the binding of maternally inherited transcription factors to the promoters of zygotic genes. So just like we saw in Drosophila, the expression of the first set of zygotic genes is activated by maternally inherited factors. Finally, another important consequence of the mid-blastula transition is that the cells acquire the capacity to move. And this is important given the extensive cell movements that need to happen during gastrulation.